Hi and hello everyone. I am Partha from the Biologist team. Would like to welcome you all for today's talk. Today we have another great personality in our platform, an eminent scientist, a leading academician, a reputed plant pathologist. For today's talk is Professor Dr. Sophien Kamoun. On behalf of the Biologist team, I would like to extend my warm welcome to Dr. Sophien Kamoun. Dr. Kamoun will be delivering a talk on Can a Biologist Fix a Smartphone? Professor Sophien Kamoun will explore this idea using a smartphone as a metaphor for living organisms. He will discuss how the capacity to read and edit genetic languages of living systems in an unprecedented fashion. At this moment, I would like to thank Professor Dr. Sophien Kamoun for accepting my invitation in spite of his hectic schedule. It's highly pride for the Biomix team for bringing your presence in our virtual platform. I also appreciate and welcome all the participants globally who joined with us for today's talk. Dear participants, we used to have question and answer sessions at the end of this presentation. You are most welcome to place your questions in the chat box of the YouTube. I and our team are here to bring your queries to our speaker to get answer out of it. And you can place your feedback as comments in the same chat box of the YouTube. Participants, please subscribe our YouTube channel to get instant notifications whenever we go live. Prior notifications for the upcoming webinar talks will be updated in the link in the description of this video. On starting our session, I am very much delighted to introduce our today's speaker, Dr. Sophien Kamoun. Professor Dr. Sophien Kamoun is a senior scientist at the St. Berg Laboratory, United Kingdom, and Professor of Biology at the University of East Anglia, England. Dr. Kamoun is known for his contributions to our understanding of plant diseases and plant immunity. He studied at Pierre and Marie Curie University in Paris and then at the University of California, where he obtained a PhD in bacteria, Xanthomonas capacis. He used genomics and molecular biology methods to obtain insights into the biology and evolution of eukaryotic plant pathogens. He also discovered virulence effect of families from plant and demonstrated how antagonistic coevolution with host plants has impacted the architecture of plant pathogen genomes, accelerated the evolution of effector genes, and drove the emergence of immune receptor networks. After his PhD, Dr. Kamoun worked at the National Science Foundation Center for Engineering Plants for Resistance Against Pathogens at the University of California, and at the Department of Phytopathology, Wageningen University, Netherlands. He was on the faculty of Department of Plant Pathology, Ohio State University, from 1998 to 2007. Before joining the St. Berg Laboratory in 2007, he served as head of the St. Berg Laboratory from 2009 to 2014. And also holds the rank of Professor of Biology at the University of East Anglia. Dr. Kamoun served as President of the International Society of Molecular Plant Microinteractions from 2012 to 2014. Dr. Kamoun is a recipient of several awards. A few are Syngenta Award in 2003, Nobel Key Award for the American Phytopathological Society in 2013, Deva Adria Prize in 2010, Academia Europea in 2011, and the European Molecular Biology Organization in 2015. He won successive European Research Council Advanced Investigator Grants in 2011 and in 2017. In 2018, he was elected as Fellow of Royal Society and he received the most prestigious Linnaean Medal for his outstanding contributions to plant sciences. With this intro, now I would like to hand over the session to Dr. Sophien Kamoun for his talk. Sir, please.
Thank you, Partha. Thank you so much for the invitation, and thank you to the team, the Biomix team. I'm really excited to share with you some about this sort of grassroots activities like yours, like this Biomics team, where there is now these online communities bringing us all together. And I think this is a fantastic development. Maybe this has been accelerated by this dramatic pandemic we're living, but it's very exciting for me that we are in different corners of the world and we can interact and exchange ideas, and for me also to interact with early career scientists like yourself and your team members. So thank you so much for the invitation.
It's always a pleasure, sir.
Thank you, Partha. I'm now going to share my screen and move to the PowerPoint. I hope everybody can see this very clearly. This is just my introductory slide. I just want to mention that I had quite a journey as a scientist. I grew up in Tunisia, my native home, but I left when I was 17-year-old. And I worked in various places in multiple continents, the US, in Europe, and I've been crossing the Atlantic back and forth. But I've always been passionate about biology. And when I grew up, I was really interested in organismal biology. I was interested in insects, actually. But my career went in different trajectory. I became more of a molecular geneticist working on plant pathogens. And then when genome biology and genomics was invented, really, I moved on to that field. But I always kept that interest in organismal biology. And maybe some of the ideas I share with you today are my feeble attempt to connect maybe different disciplines of biology, the molecular sciences, which focus a lot on mechanistic research, and maybe organismal biology, which is more interlinked with evolutionary theory, with evolutionary thinking. And I have a feeling that there is a bit of a gap in our biological life sciences where the evolutionary community doesn't always integrate the ideas from the mechanistic research, from the more molecular mechanism, and vice versa. I think the molecular mechanism people don't not to really integrate evolutionary biology. So this this talk really starts with this paper that is a very influential article, a very influential commentary written by Yuri Lazebnik back in 2002. Personally, I only read this about 10 years ago. I wasn't aware of it, but this paper is viewed as a sort of a founding manifesto for systems biology. And the argument that Lazebnik was making is that biological system can be viewed as radios, and where you have basically an input and an output. And then what systems biology should do really is try to certain signal is basically expressed as a particular output in the same way that radio would function. So I have to say that this article is very influential. It's is a lot of the argument that Lazebnik make are absolutely sound, and he emphasizes, for example, the complexity of biological systems and how you really need to have some sort of systems approach to understand how living systems function. And this really has been integrated in a lot of the systems biology thinking. And this is an example. This is a slide I borrowed from a lecture on systems biology and network biology. And these are very valid arguments and very valid thinking. And I think especially our colleagues who came to biology with a physics background, they really appreciate this angle to biology. But I have to say that when I read this article, I was left wanting, because my view of biology is a little different. I understand the mechanism. I appreciate all this thinking about essentially unraveling all these interactions within a certain signaling circuitry and so on. And I understand the limits to the approaches that biologists usually take in studying biological systems that Lazebnik put together. But I thought there was two things that were missing in his metaphor. And I thought these two actually are really fundamental aspects of biology, so we cannot just ignore them. And I thought the radio metaphor was missing those. The first one is that biological systems are defined by this very clear separation between what is written in the genome, what people like G.C. Williams, for example, call the codical domain, the code essentially, which is written in the genome, which is written in the DNA, and then the hardware or the organism itself. This is really fundamental in biology. Biological systems are based actually on the code, and that code is written in DNA, and that's a separate entity from the organism itself, how that code is actually translated into an organism or into a living system. So that's the first thing that the radio metaphor really ignores, and this is truly fundamental in biology. And these are things, this concept have been, I think, in my view, best really written about by G.C. Williams, but also Richard Dawkins' views in the selfish gene are quite similar to this. And then the second point, and I think, again, that's another fundamental aspect of biology, is that biological systems are not optimally designed. They are not. They are suffering, if you like, from historical historicity. And we'll talk a little bit more about that. But the point here is that one thing that differentiates biology from, let's say, design, or even sometimes physics, is that biological systems have evolved. So they are linked to that continuous chain of heredity to some ancestors. And that has huge influence on how biological systems are today. And also that is resulting in an extraordinary messiness of biological systems. Biological systems are innately messy. And so they really differ a lot from designed systems. What this means in practical terms. But I thought these two fundamental aspects of biology were sort of ignored to some degree by the radio metaphor. So I decided a few years ago to write about this. This was kind of in the back of my mind ever since I read the Lazebnik article, and I thought, okay, how best to make my point is basically to use another metaphor. And I thought the reason why the smartphone is a better metaphor is because the smartphone has also that differentiation between the software code and the hardware. And so if you think about it, the smartphone, because of that feature, because you have the software being written in a particular code that is really a distinct entity from the hardware of the phone itself. Because of that, perhaps smartphones are a slightly better metaphor for biological systems than the radio. So what I will do really is I'll take you through those ideas I developed in that article, and the metaphor I wanted to have is let's think about aliens with extremely advanced technologies who land on Earth and get intrigued by smartphones and decide to study them and understand them in the same way that biologists really study living organisms and living systems. So let's assume these aliens have extremely developed methods and technologies, and they can actually decipher the smartphones in all its detail in a much better way than we can actually do today with biological systems, the smartphone, and understand the features of the smartphone. And so one aspect that the aliens would definitely struggle with, even if they have extremely advanced technology, is this question of why are the keys or why are the letters in the phone, in the keyboard, arranged in this format, the so-called QWERTY format, the QWERTY. And I think it's very clear that the aliens would really struggle with understanding that. Now, if you think about like in the same way that biologists approach biological problems, the biologists might start thinking, well, maybe this is adaptive. Maybe there's an adaptive feature to why the letters are organized in this way. And they might do all kinds of experiments trying to demonstrate that there is organized this way. in reality, if you think about the QWERTY keyboard, the reason why it's organized like that is very complicated. There's a lot of different theories. Some people actually argued that the reason the keyboard is organized in the QWERTY fashion is to avoid jamming the old typewriters. That theory has been disputed, but that's interesting, by the way, because that would mean that the QWERTY keyboard is really was designed for a feature that is useless today in the modern smartphone. But there's another theory, which is, I think, the most likely, this format of the first popular typewriter that became popular. And once people start using that keyboard, then it was very hard. Then all the other companies that built typewriters just kept using the same keyboard, and so on and so forth. But you see my point, I think, that the reason the smartphone keys are in the QWERTY format is simply because of a historical reason. It's because the first typewriter, something that is long gone, had the QWERTY keyboard. And because of that historical constraint, all the letters remain organized in that way, and we still use the QWERTY keyboard in things like the smartphone. Incidentally, the QWERTY keyboard is not the best one. There's a so-called Dvorak keyboard. I did not actually know about this when I wrote the article, so I discovered this only recently. And this is organized differently. It's AOE, as you can see here. It's different from the QWERTY keyboard. But the point is that this keyboard allows you to type faster. So this is an optimally designed keyboard. And some people use it, so it is out there, and you can order, if you like, this keyboard for your computer. But as you can imagine, this is not widely used. And again, this helps illustrate the point that the historical constraint resulted in a suboptimal solution being retained in today's keyboard. And I can imagine even with smartphone that actually improving the keyboard will be a huge benefit, but we're sort of stuck with that history. Okay, so how do we crack that problem? How would the aliens crack the problem of the keyboard? I think this is a very important point in biology. That's really time cracking the problem of the keyboard by studying a single phone or a single keyboard. They'll need to do essentially a comparative analysis of a population of smartphones from as many different regions of the world as possible. They might discover QI, and they might find that there are reasons why it's different. Some of them are a bit adaptive because that keyboard is more adapted to the French language. They'll really, to have an insight on this, they might actually go and find some antique store where that antique store would be selling typewriters that nobody uses, and they might realize then that these are relics, maybe almost extinct relics of past usages of these keyboards. And then by sort of doing that comparative analysis and reconstructing the history of what happened, how did the keyboard become what it is today, they will have actually more of an insight into understanding this problem than by studying a single keyboard, even if they can measure all the electronic components and so on, let's say typing speeds and also all kind of stuff, and try to understand how they work. But really, you need the comparative approach to crack the problem. So the message really from this is that we need an evolutionary perspective. Because without an evolutionary perspective, our mechanistic understanding of biological systems is unlikely to innately messy, and we do need that evolutionary perspective to really get that insight, to get the true understanding of the mechanisms. Let me use another example, but now a real example in biology. I mean, let's say penguins, right? So penguins have wings. They're viewed as being flightless. They're flightless, but they do have wings. And if you're studying only penguins and you try to understand what are wings for, what is the function of wings, you'll make some progress, because you'll probably sooner than later realize that penguin wings are actually very good for swimming, and they're good flippers for allowing penguins to swim. They're not necessarily very valuable on land, but you'll be probably very happy writing publications about how penguin wings are important for swimming, and how this is that you have actually now figured out the function of penguin wings, and you might even speculate about wings in general. But you can see the point here that if you really want to understand wings, you need to have the comparative approach, because if you studied birds from the comparative angle, you'll have a much better, much deeper understanding, and a much more sophisticated understanding of why penguins have these wings, and why wings are structured this way, and why there are modified feathers and so on, than if you just study penguins. Francis Crick, actually, in his wonderful book, really eloquently wrote about this by saying that biologists must constantly keep in mind that what they see was not designed, but rather evolved. And and I totally share that view that's in that second quote here, is that you would expect really that evolution would play a large part in guiding biological research, but sadly that's far from the case. So I do wish to see a better integration of evolutionary thinking and evolutionary biology into biological research, especially into mechanistic research. And that's something that we are trying to do actually in my group in studying plant pathogen interactions and plant immunity. So that was my message about historicity, and I hope you can appreciate why this is important, and that the evolutionary approach really does bring another dimension to your mechanistic understanding of biological systems. But let's pause also and think where we are now in 2020. So we're 20 years into the 21st century, and it is very evident that biology is really blooming in this century. And I cannot even imagine where we will be in 20 years or in 40 years or towards the end of this century in terms of our capacity to not just understand biological systems, but also modify them. And that's really driven by two technologies. One is genome sequencing, which has really matured now to point where it's really impressive. There are still some advances there. We still have large complex genomes that are still expensive and so on to access, but we're getting there. And in a way, if you think of the genome again as a software code or as a book, what that allows us to do is to read the colorful. And in the book, metaphor would be equivalent to word processing, which is the capacity to edit genomes. And this was available already for a few years ago, but the capacity to edit genomes is something that really sort of went popular through the CRISPR technology that I will talk about in a second. So the point is really here is that in the 21st century now, biologists have the capacity to read and edit genomes. And this means that we can access now the codical domain of living systems in a really unprecedented fashion, in a very powerful way. So now the question is, if you want to really go back to that software, to that smartphone and software metaphor, then what does this mean? Well, essentially, now a lot of biologists became software engineers, where we can actually go into the genome, read the software, and modify it if we wish. And modifying the software is extremely powerful, because essentially it's like writing apps. So who is more powerful in terms of the smartphone? Is it the software engineer who writes a new app for the phone and allows you to do absolutely amazing things, almost like magic sometimes? Or is it really the hardware engineer who can modify maybe the screen or modify component, etc.? So it's really the software engineers are extremely powerful. I mean, both are needed, obviously, there's no question about it, but the software engineers are the ones that can really unleash their creative power in terms of exploiting the potential of a smartphone. And as I said, really, some of those applications almost feel like magic. They're so powerful. So essentially, geneticists or genome editors, if you like, bioeditors, we can call them, in a way, they are operating a little bit like software engineers as far as the smartphone is gone. And so essentially, they can really modify even hack biological systems, sometimes without really an understanding of the system itself. If you think about it, the software engineer, they don't necessarily need to understand all the electronic machinery of the phone to write very powerful software or very powerful apps. So that's really important aspect of the metaphor, because as genome editors, sometimes we can remove a gene or modify a gene and then obtain an output that is quite powerful. But that doesn't necessarily mean that we understand all the complexity that's behind the system. So if we go back to, again, that thinking about systems biology, yes, it would be great, of course, to have that understanding. That won't hurt, and that would allow us to make better changes and better modifications. But in many cases, we can actually modify the system that does not, without a deep understanding, that does not necessitate actually the deep understanding of the complexity to be able to actually do something quite powerful. So let's talk a little bit about CRISPR, and I'll switch a bit about also maybe give you an example of some of the research we do to relate it to what I was talking about. I think I suspect you have heard all about CRISPR, which is a very recent discovery and a very recent Nobel Prize, which was awarded just last month to these two wonderful ladies, Jennifer Doudna and Emmanuelle Charpentier, who received just 7-8 years after their discovery that allows you to edit any sequence in the genome in pretty much any organism these days. That is not the first time gene editing was done, but it just made it easy and it made it accessible to pretty much any biologist with a molecular biology lab in the world. I don't want to get into a lot of details, but the point here is that it's a programmable nuclease that allows you to make cuts that are guided at particular sites in the genome. And because the programmable bit is an RNA, shows in the genome of pretty much any organism. So literally the Nobel Prize winning paper came out in August 2012, and within months there was already applications of doing gene editing in human and other organisms. So this is you can see here, this is January 2013, so literally half a year after the CRISPR paper came out, there were the first applications for human gene editing. And then just a few months after that, these are the first examples in plants, and that is, again, really fairly easy technology that is quite powerful, allowing us to edit plant genomes with many applications. As you can imagine, this is really powerful technology that allows us to modify plant genomes, modify the software of plant genomes, if you like, the software code of plant genomes, the DNA, in a guided way. And that can really accelerate plant breeding, accelerate the improvement of our crops, for example, in a very powerful fashion. So we got involved in that as plant pathologists. We're interested in using this technology to make plants resistant to pathogens. This is a technical slide, but I just wanted to point to, for those of you who are interested in getting more details, that you can look at this review. And this is really our version of the Lazebnik circuit for plant pathogen interactions. We have a good understanding now that pathogens secrete molecules that modify the host plants, and then plants fight back using immune receptors that can detect this pathogen. And this complex interaction is understood, but we just know the components. We know the components that are involved. We don't necessarily know how they mechanistically work together. Most of the time, we actually don't really know how these different components function. But genetics and the capacity to access the genome has allowed us to discover a lot of the players, a lot of the parts that are involved in these pathogen interactions. For example, the receptors, the negative regulators, and so on. So then as editors, we can actually go and modify these genomes and make these plants resistant to pathogens. So this is an example of a CRISPR crop, if you like. This is a tomato where we removed a gene called MLO. We just removed a piece of text, so that's what you can see there. So that's on the top bit is the sequence of your regular, as we say, wild type tomato. And then the different plants underneath reflect the different mutations we introduced. So essentially we removed 48 or 49 letters from the genome of tomato, from the software code of tomato, perhaps. And by removing that, we disable this one gene MLO. And by disabling that gene, we can make the tomato plant resistant to this fungus, to this fungal pathogen. That's what you see there on the bottom side. The plants on the right have that mutation, they have the deletion, and they're resistant to this fungus infection. I'll be honest with you, we don't fully understand how this MLO protein functions. So our understanding is quite limited in terms of how this protein confers resistance, but we know it works, and we know there are no detectable side effects in terms of yield and so on that are associated with this mutation. So that brings me back to that first point I made, that really we can actually modify and hack biological systems, perhaps, without a deep understanding of the complexity of the system. So we can even make a tomato plant, resistant to fungus, without really having that intricate understanding of the system, as Lazebnik was alluding to, that we really need to understand how a signal is translated into an output, and that understanding is absolutely critical for being a biologist. Actually, we can do this without a particular deep understanding of the complexity of the system. And that's really why genome editor really, in a way, are operating perhaps in editing biological systems. Okay, so just to conclude here is the point I want to make really is that what we need to do and what we need to know is how to interact with the software to create new apps. And that's really what gene editing allows us to do. So essentially, biologists now are operating as software engineers who can hack and write applications to modify these genomes. And this is an important point. It's of course, it doesn't hurt to have an understanding of the complexity of the system you're modifying, but this is not necessary to make a beneficial modification. And that's, I think, a really important point I want to make here, is that doing bioediting, modifying this genome, does not necessitate a full understanding of the complexity. Statement taken from the article, and I think it's worth it that we go through this together, because in a way what gene editing is, it's kind of a new evolutionary force. It's accelerating a process that has been taking place for eons. I mean, obviously mutations and modification in these genomes are continuously happening. As you hear now, for example, with SARS-CoV-2, with the coronavirus, that virus is mutating. That's normal. That's perfectly natural. What would be really strange is actually if it didn't mutate. That would be unusual. Biological systems are continuously mutating, continuously changing. And what gene editing does, it's simply accelerating this process that has always been taking place. And that is totally integral to biology. And actually a basis of the messiness also of biological systems, because they're not sitting idle. They're continuously changing and mutating and evolving. However, I think what we really need to do here is to integrate evolutionary biology, evolutionary research. And I think the power of gene editing will become more evident when we comprehensively understand this tinkering nature of the evolutionary process, and also all the issues related with that tinkering, because tinkering comes with trade-offs. The systems are not optimally designed, so that means they have trade-offs. And also it comes with constraints, and those constraints are also important to understand. So really we need to do a better job at integrating our mechanistic systems and theory. And hopefully this combination of gene editing with a better appreciation of evolutionary biology, this will really truly cement the position of biology as an information science in the 21st century. So that's what I had to present. I wanted to thank the folks who pioneered the gene editing, the plant gene editing work in the lab. It started by Vlad Nekrasov there, and he shared his vectors very early on through this platform AdGene, and they were shared by over 1,000 times. So it's really proud of contributed to gene editing research. So I think I'll stop there. I want to remind you that this year is the International Year of Plant Health. That's what the United Nations has declared. Unfortunately, it's been hijacked. It feels like it's been hijacked by another pathogen. but I still want to remind you the importance actually of plant pathology and plant health in the 21st century, and also the promise that gene editing brings to addressing plant health issues. So with that, I will be very happy to answer any questions. Thank you for your attention. And I understand the questions will be typed in the chat, right, Partha? So I don't have access to the YouTube.
Okay, Zero. Yeah. Thank you so much, sir, for your lecture on Can a Biologist Fix a Smartphone, which emphasized to read and edit the genetic language encoded in the genome that manner. I hope there are so many participants, enthusiastic participants throughout your talk, and we have some questions, a few questions from them. With your permissions, I will forward these questions one by one to you. These questions are very basic questions, since basically people are from biological background. They have too little ideas about domains of codical languages, and they are asking about different codes in smartphone and how we crack it.
Yeah, that's a very good question. So the question is, how do we compare different smartphones and different codes? And I'm trying to see if I can access the YouTube here, but okay, let me mute this. So 1 second, sorry, just so it doesn't interfere. Yeah. So so it's that's like I said, there are two technologies really that completely transformed biology, the revolutionary technologies in biology. One is the capacity to read the genome. So some of you know that, but for those of you who aren't biologists, then basically the genome is DNA, form of DNA, and the DNA is formed of 4 letters. And essentially, that's like a book. If you think about it, the metaphor of a book is perfect for that. So the book is basically a series of letters. The genome is a series of letters, in the same way that the software code also is a series of characters. So the first technology that started really at the end of the last century is the capacity to sequence the genome and to basically to read that code. So that's the first technology. The second technology is the editing technology, which is basically like modifying the text and just like you would do with the word processor. But the first technology, so imagine you have 2 organisms. So even, for example, Partha and I, right? We can have our genome sequenced and then being compared. And then we will see the differences between our genomes. And that may or may not give you some insight about what we are biologically different. Maybe I have some I don't know, some gene mutation for diabetes that Partha fortunately would not have, and then that you'll be able to find out the difference between us by through reading the genome. So that's what people do, in the same way that if you compare the 2 versions, that's what biologists would do if they were studying smartphones. They would compare the different software codes and then try to understand why a certain app is working in one way versus the other. So that's really what the technology of genome sequencing, of the field of genomics that you hear about in the news is about. It's about reading the code and comparing it. A lot of genomics is really comparative analysis.
Yeah, sure, sir. The next basic question is, can you put what is a codical domain in such a way a biologist can understand?
Yeah, so to be honest, when I wrote this, there's a lot of discussions among us about what the codical domain is. And I really encourage a well-written article or a chapter in his book that I cite in my article that is discussing this topic. So if you want to know more, I would check that. But essentially, the best way to think about it is it's really the information that is coded in the genome, okay? So that's really what the codical domain. Think about this way, the sequence, the DNA sequence is information. And that information is the code. And that code is really the code for essentially building the organism, right? So that's what I mean by the codical domain. It's better to actually think about it as an information rather than a physical entity. Of course, it's coded in the DNA, but in reality, the power of a codical domain, it's irrelevant how it's coded, if you think about it. Like, for example, we actually, as biologists, as geneticists, we transform the genome sequence from DNA into letters that are in the computer or even on a piece of paper, right? And that's still the same information. So if I tell you the sequence is ATCG, ATCG, then you can understand what I'm meaning, because the information that matters. So the codical domain, as far as biologists really should think about it, it's really the information that is coded in the DNA, because it is information. You know, in the same way that, again, as language is information, like I could give you the word now information, or I could write it on a piece of paper, I could type it in a computer. The information doesn't change. It's just how it's physically kind of described that changes. So DNA really codes for information, and that information is the codical domain. In the same way that in the software code, the software engineer will type some code in a particular language, and that code is actually information that is then translated by the smartphone or by the computer into some actions. And that's the same way that the codical domain, the cell, works. It's transforming that codical domain, it's transforming the information in the DNA into some concrete actions like synthesis of a protein and so on. So I hope this is clear, but it's a little bit a philosophical point, but it's very important, I think, that we realize that biological systems have that information coded in them. And that's a very important aspect, a very important feature of biological system. That information actually is what is transmitted also from generation to generation, that is modified through mutation and so on.
Yeah. The next proceeding question is that you're motivating us to control living organisms, like we control hardware through coding and applications.
Sorry, can we do what? I missed that. I'm sorry.
Yeah. Motivating us to control living organisms like we control hardware through coding application software.
Controlling, okay.
Do you think living organisms would be acted if tools and techniques keep advancing? We advance research in the field of genome editing.
Sure, that's a very good and very deep question, and it's really beyond me and beyond this discussion to address all the ethical issues of gene editing. Obviously, with human gene editing, I'm personally, as not just as a scientist but as a citizen, I guess, I'm against germline editing in humans. I think that's unacceptable at this point of our understanding and our appreciation of the ethics of biological modifications. I'm against modifying the germline of any human being. I think that would be inappropriate. Modifying, for example, let's say blood cells to fix a certain mutation or so on, and then re-injecting them in somatic cells, as we say in biology, I think that's understandable, that's therapeutic. But modifying the germline, I think that's probably the limit right now. But let's step back. Let's not think just about human applications, where there are clear applications of gene editing for as therapeutics for certain diseases. But let's think about agriculture and crops. So I want to step back. I don't want to think about gene editing without the context of what agriculture is and what crop domestication is about. We have already modified the plants we grow and eat in a very dramatic way. You'd be hard-pressed recognizing any of the domesticated crops we have in the wild. They basically don't exist anymore. We changed them over thousands of years in such a way that basically they are very hard to distinguish from their wild relatives or wild ancestors. So the way to think about our crops really is that they are domesticated, and they are really, in a way, co-evolving with us now. Many of them actually cannot survive without human care. They would not survive as wild plants. People keep cats or dogs and so on. So this is text of looking at gene editing. Now, after that, what is acceptable, what is not, again, this is beyond this discussion and beyond my own expertise, really. Ethics is a very complicated topic. But we should really appreciate that, when you're eating rice or eating maize or potato, these are not wild plants you're eating. You're eating actually something that has been modified already through thousands of years of domestication. And that's really the context we have to work in. These plants are already essentially made dependent on humans and made dependent on farmer care to basically nourish us. And it's a form of co-evolution if you think about it, because they're also benefiting from us, right? Because in a way they are actually using us to take care of them and grow them and cultivate them and even protect them from pathogens and this sort of things. So it's an interesting case of co-evolution.
Yeah. The next interesting questions from participants is, what is the unforeseen risk of producing the mutation in crop that will have an influence on human beings in a long run of consuming the particular genetically modified organism or a crop?
Yeah, so the question is about the risks of technologies. I think it's a very important and very deep question. I think it's really difficult to give a catch-all answer to a question like that. The reality is that it's really a case by case. I think the point I would make strongly, with a lot of confidence as a scientist who understands this technology, is that the technology itself is not particularly dangerous. There's nothing inherently dangerous in doing genetic modification, because this is, again, something that is happening in nature that we have been doing through using other methods throughout the years. What happens with gene editing is that it becomes more precise, more informed, more specific. For example, we have a lot of the crops we eat, almost all of them are the products of mutations that is often driven through human intervention, either by doing the mutagenesis or by selecting the mutants out of a larger pool of plants. So I think inherently gene editing is not different really from what happens in nature and what has happened throughout crop domestication. Then I think the best way to answer it in a reasonable way in terms of its biosafety and so on. The risk that there is a mutation that will be sort of a Frankenstein mutation, I cannot visualize a scenario like that, how modifying the genome will create some sort of risk for humans. Maybe it can increase allergenicity, for example, but then again, you would want to test for that. You will want to experiment for that. You want to have reasonable science. For example, we are introducing and moving entire genomes of organisms, like, for example, the virus genome, like the coronavirus. Again, I go back to that. That's actually an entire genome that has been introduced really throughout the world. But we are also introducing all kinds of crops and pests, all the time, by shipping plants from one organism. That is causing a lot of damage, actually much more dangerous right now than modifying genomes. So, and in that case, what you're doing really is a form of genetic modification, because you're introducing to a country, for example, an entirely new genome of a new organism that hasn't existed there before. So I think the technology needs to be regulated like every other technology, but I don't think it's correct to say that the technology is inherently dangerous. I think it's really the output that needs to be evaluated rather than technology itself.
Yeah. The next question also based on, will it be really used in ground level or it is just experimental?
In terms of generating useful applications in several systems. So I think no, that's already happening. There are many companies that are developing in agriculture, for example, CRISPR-based mutations. There's also gene editing using other methods than CRISPR, but the beauty of it is actually even a lab like mine can generate new mutants, like the tomato I showed you, the fungal-resistant tomato. That's something we did in our lab in about 10 months. So that's actually the beauty of CRISPR more than anything, is that it is fairly easy to implement. I don't need to be a large international corporation to generate this mutant, and I think it's accessible to labs, for example, in India and Bangladesh. We have collaborations throughout the world with many labs who are using these vector systems for CRISPR. So it's really a technology that is quite relatively easy to access as long as one has a molecular biology lab. So no, it's happening already. It's not a theoretical exercise.
Yeah. The next question is that, is this possible to use siRNA technology to combat against COVID-19?
Question. That's beyond my expertise. Honestly, I wouldn't know how to answer that question. I'm not a COVID-19 expert by any means. I think we can be very excited about the developments about the vaccine therapy for COVID-19. I've been following that field quite closely, and it's quite exciting to see that the first results coming out of the vaccines are promising. Let's hope that this will continue also with maybe the more standard versions of making these vaccines, which I understand, for example, in India, there's a massive effort to also make a COVID vaccine. I think that's going to be hopefully the way out of this crisis. And I hope everyone is staying safe, but I don't know how to answer this question. I'm sorry.
As a follow-up question, will CRISPR might help to discover a vaccine for this pandemic?
I don't think that's needed. I think CRISPR is a different technology for that. The vaccine technology is actually, fundamentally, in principle, is not very complicated. It's if you think about the virus being multiple component, and you may have heard about this spike protein as being one component of the virus. What basically taking that spike protein, separating it from the rest of the virus, and then delivering it to the human body, and then what happens then? Our immune system will recognize that spike protein as being a foreign invader and will make antibodies against it. But the spike protein on its own cannot do any harm. It needs to be part of the entire virus. So separating that piece of the virus and injecting it as a vaccine in various methods is how vaccination occurs. And that technology does not require CRISPR type technology. There are other molecular biology tools that are used, but not CRISPR. But yeah, that's the principle of vaccination essentially.
The next question is that what is the contribution of artificial intelligence or data sciences in the field of genetic engineering to help us advance intelligence or data sciences in the field of genetic engineering to help us advance?
Also, the impact of AI or on biology will have a huge impact because, again, biology with genome sequencing, with the genomics revolution, became a data science. It's basically an information science now. So biology can be viewed really as an information science, and therefore all the tools and methods that were developed for all the types of information science, all the types of information technologies, they become applicable to biology. But we're really at the beginning. And I think one challenge we have in biology is not necessarily collecting the genomics data now anymore, the code, if you like. It's actually collecting the other information that are complicated. And also, for example, if you grow a plant under different conditions, it will look totally different. So that's a challenge for what we call in biology phenotyping, which is really collecting the output information in a way that you could link to the genes and to the genotype. And that's something I think that's probably one of the first application where AI or machine learning I think is starting already to have an impact in biology.
Yeah. So let it be the last question before you leave a lab from India as well as across the world.
Well, I think that's a great question. Thank you so much for asking it. I think I would encourage any candidate to look at our websites and look at the different programs we have. But I want to take this opportunity to announce that my institute, the Sensby Lab, will start a master's program in next year, in 2021. And that program will be open to anyone, anywhere. And we will have also quite a generous number of fellowships to support that. So at least at the master's level, there will be really good funding next year to host students, hopefully from India and from anywhere else. Otherwise, do check the website and check the various advertisements we have for PhD positions or for other positions. That's the best way to approach it. And I wish you the best of luck, and thank you for the question.
Yeah. Thank you so much, sir, for your time and patience in listening and answering all of our questions. I hope all participants are satisfied with the speaker's elaborate explanation and answering all your questions. Thank you so much, participants. And I thank speaker of today's talk, Dr. Sophien Kamoun, for spending your valuable time with us. I extend my sincere gratitude to you on behalf of my entire team. Thank you so much, sir.
Thank you, Partha. Thank you, Biomics team. Appreciate the invitation.
Gratitude to you on behalf of my entire team. Thank you so much, sir.
Thank you, Partha. Thank you, Biomics team. Appreciate the invitation.
Yeah. Once again, I thank everyone who joined with us for today's talk. Few announcements to the participants. Participants, please subscribe our YouTube channel to get instant notification whenever we go live. Prior notifications for the upcoming webinar talks will be updated in our Telegram channel of the Biomics. You can get access to the Telegram channel, link in the description of this video. Stay connected with the Biomics team for further updates. Join with us. Stay tuned. Thank you. Thank you all. So thank you so much, sir.
Thank you. Thank you, Partha.
Bye.
Bye.
Thank you. Have a nice day.
You too.
Bye.
Have a great day. Thank you.
Thank you, sir.